Hi everyone. In this lecture, we will be talking about the cell cycle. Now, what is the cell cycle? This is the essential mechanism by which all living things reproduce. And to put it simply, this is the process of cell duplication, where one cell becomes two cells. Here we can see the three main processes that occur in the cell cycle. First, we have to have cell growth and chromosome duplication. After the chromosome has been duplicated, there is chromosome segregation, which is the division of the chromosome into two equal parts. Then finally, we have cell division or cytokinesis, in which one cell becomes two daughter cells. Now, how long does it take for our cell to divide? For some embryonic cells, it can be as little as a few minutes. Here we can see that for early fly embryo cells, it only takes 8 minutes for one full cell cycle to occur. Meanwhile, for differentiated cells and for higher organisms, it can take a few hours. In this example, we can see that intestinal epithelial cells can take as much as 12 hours, and for fibroblasts, they can take approximately 20 hours to divide. Meanwhile, for some terminally differentiated cells like neurons and muscles, those types of cells no longer divide. Now, the eukaryotic cell cycle occurs in two main phases. The first one and the most exciting one is the M phase, which is composed of mitosis or nuclear division and cytokinesis or cytoplasmic division. The M in the M phase is actually for mitosis. After the M phase occurs, we have the interphase. Now, although the cell looks relatively inactive during this phase, there are still many processes that occur within the cell. First, we have the S phase or the synthesis phase, and this is where DNA is replicated. We also have two gap phases, G1, which occurs in between the M and the S phase, and G2, which occurs in between the S and the M phase. And the G here stands for gap phases. In these phases, the cell continues to grow and gather nutrients in order to ready it for the incoming M phase. Since the cell cycle is a very important process in the cell, it also has a cell cycle control system. Now, in animal cells, this robust system is composed of a network of regulatory proteins. These are the different purposes of the control system. First is to allow the cycle to occur in a set sequence. This means that the M, G1, S, and G2 phase have to occur in specifically this order. At the same time, this system also ensures that each process has been completed before the next one can begin. In cases where this system is not followed or this process is not followed, this might lead to abnormalities in the daughter cells. For example, if the cell has just undergone mitosis and does not undergo the G1 phase before it gets to the S phase, that means it goes from M to S phase directly, then it might not have all the necessary building blocks in order to successfully replicate its DNA. And as a result, the daughter cells would have incomplete DNA. Now, in order to check that all is going well, our cell cycle control system has checkpoints, and these can be used to pause the cell cycle at certain transition points. So in cases where the cell can detect that there are some abnormalities, it pauses the cycle so that it can try to fix them. For example, between the G2 and the M phase, the cell has to check if all the DNA has been replicated and if any DNA damage encountered by the cell has been repaired. If it sees that these two questions have already been fulfilled or answered, then it can enter into the next stage, in this case, mitosis. Just like any other pathway in the cell, there are also some molecular switches that are involved in controlling the cell cycle system. Many of them are controlled by phosphorylation and dephosphorylation of proteins. The main protein involved in the cell cycle control system is what we call cyclin-dependent protein kinases, or CDKs for short. This is the major protein kinase. Again, a kinase is a protein that phosphorylates other proteins or transfers a phosphate group to them. Now, the CDKs are present in all cell phases of the cycle. And here we can see an example of our cyclin-dependent protein kinase, or CDK. So even though they are present in all phases of the cell cycle, they can only be activated at certain times in what we call a cyclical pattern. So there are times when they are activated and there are times when they are inactivated. And the way that they are activated is by another protein called cyclin. And together, they form the cyclin-CDK complex. So without the cyclin, even though the CDK is present in the cell, it cannot function. That's why it's called a cyclin-dependent protein kinase. In this table, we can see the different cyclin-CDK complexes that are involved in the cell cycle. For each cyclin-CDK complex, there is a specific cyclin involved and a specific CDK partner. But for simplicity purposes, we simply call the cyclin as whatever phase of the cell we can find them in. For example, cyclin D is also known as G1 cyclin, cyclin E is also known as G1S cyclin, cyclin A is S cyclin, and cyclin B is M cyclin. And here we can see the different purposes of our cyclin-CDK complexes. G1-CDK is the complex used to drive the G1 phase. That means it allows the G1 phase to proceed as normal. And then we have the S-CDK and G1S-CDK complexes, which launch the S phase, and M-CDK, which launches the M phase. As we mentioned earlier, our CDKs have a cyclical activity, and this is activated by high cyclin concentration. So here in this figure, you can see that our cyclin-CDK activity, in this case the one being shown is M-CDK activity, is only present at certain times of the cell cycle. And these coincide with the high concentration of your M-cyclin. So here we see that as the M-cyclin concentration increases, at its peak, this is where we see the M-CDK activity happening. In this figure, we can see that active cyclin-CDK complexes are the ones that actually move the cell cycle forward. Here we see an example of two cyclin-CDK complexes. We have S-CDK and M-CDK. And here we can see that S-CDK drives the cell towards S phase, meanwhile M-CDK drives the cell towards M phase. Another thing we see is that these complexes are only active when cyclin concentration is at its peak. So for example, for S-CDK, it is only active when the S-cyclin reaches its peak, and the same can be said for M-cyclin here at its peak. Now, another thing we'll notice is that the cyclin concentrations gradually rise and abruptly fall all throughout our cell cycle. Here we see that the S-cyclin slowly increases, and here the M-cyclin is slowly increasing as well. But after they have served their purposes, then the levels abruptly fall, and this ensures that the cell is able to stop the cell cycle phase very abruptly. Now, how does the cell regulate its different cyclin concentrations? There are a few strategies, and the first one is the direct destruction of cyclin. So when cyclin is no longer needed, the cell uses a protein called the anaphase promoting complex or cyclosome, abbreviated by APCC, and this tags our M and S cyclins for degradation through ubiquitination. Ubiquitination is the addition of a ubiquitin chain to certain compounds or organelles in the cell, and usually the addition of ubiquitin is the marker for the cell to destroy whatever it is attached to. By destroying the cyclin, this leads to the inactivation of the cyclin-CDK complex, and we can end the mitosis when that happens. The next strategy that the cell uses in order to regulate cyclin-CDK activity is with the use of inhibitory phosphates. Normally, phosphates are used to trigger or to turn on certain proteins, but in this case, they are used to inhibit the cyclin-CDK complex. And the one being shown here is the M-CDK complex. The protein that places these phosphates are called inhibitory kinases, and they inhibit or inactivate your cyclin-CDK by adding our different inhibitory phosphates. The one being shown here is called WEE1. In order for this inactive complex to become active, another protein has to remove the inhibitory phosphates, and that's what we call the activating phosphatase. Once it has removed the inhibitory phosphates, the cyclin-CDK complex can now become active. The specific activating phosphatase being shown here is called CDC25. Lastly, the cell can also inactivate the complex directly with the use of CDK inhibition proteins. These proteins can act in two ways. The first is to block the assembly of your cyclin-CDK complex. So it doesn't allow these two components to meet each other. Another way that it does this is by blocking the activity directly of the formed cyclin-CDK complex, and that is what is seen in this figure here. The inhibitor protein's name is called P27, and here you can see that once it binds to this cyclin-CDK complex, it is no longer active. Now, the different methods that was used by the cell to regulate the cyclin-CDK activity can be used to create pauses in the cell cycle at the certain checkpoints, which we discussed earlier. These pauses can occur due to a variety of factors. For example, if the DNA replication was not complete, or if there's DNA damage, or maybe the chromosomes did not properly attach to the mitotic spindle, or if the environment is not really favorable for cell growth. Then the cell can pause the cell cycle by inactivating the cyclin-CDK complexes. And the mechanisms it can use can also be varied, and some examples were given in the previous slides. Let's now move on to specific examples of starting and pausing the cell cycle at each phase. First, we will start with the G1 phase. This slide is showing the different events that occur in the G1 phase. First, we have to have the inactivation of our different cyclins. In order for a cell to proceed to the G1 phase, the M phase has to be stopped. And here, the existing SCDK and MCDK complexes are inactivated. At the same time, there is also the prevention of synthesis of the new cyclins. After this, the cell has to be stimulated by mitogens in order to start preparing for division. Mitogens are extracellular signals produced by other cells. And what these do is that they stimulate the synthesis of G1 and G1S cyclins. At the same time, it kickstarts DNA synthesis and chromosome duplication. Once it has been stimulated to start dividing, the cell will start its cell buildup, wherein they will gather the necessary components for cell growth. In this figure, we can see one example of how mitogens work in the cell. The specific example given here is the effect of mitogens on the protein called the retinoblastoma, or the RB protein. Normally, this protein represses genes for cell proliferation, and we can find it in all nuclei. So as long as this protein remains active, the cell cannot move into the other phases of the cell cycle. Now, once the mitogen has been detected by its receptor, its effects is that it activates the G1-CDK and G1S-CDK complexes, and in turn, these complexes inactivate our RB protein with the use of inhibitory phosphates. Now, once this protein has been inactivated, the transcription regulator that it was blocking can now transcribe the genes needed for the cell's entry into the S phase. In this figure, we can see one example of how we can pause the G1 phase, and that is with the use of your P53 protein. This protein is present in the cell whenever DNA damage is present, and what it does is that it promotes the production of the CDK inhibitor protein P21, which we discussed in a previous slide. Now, as long as the P21 protein is present in the cell, the G1S CDK and SCDK complexes remain inactive, so the cell cannot proceed to the S phase. If there is enough P53 protein that is being produced by the cell, this means that there is too much DNA damage for the cell to deal with. It will lead the cell to commit apoptosis or cell suicide. Now, this protein is incredibly important because the absence or defect of the P53 protein and its gene usually lead to the development of cancers. And in fact, almost 50% of known cancers have this defect in the P53 gene. Next, let's move on to the S phase. These are the two events that happen in the S phase. We have the replication of DNA. Here, the genome has to be copied once and only once. After the genome has been copied, the cell can now move into the G2 phase, and this is usually continuous after the S phase. In many cells, once they have gone past the G1 phase, the S phase, G2 phase, and M phase actually go in a very, very fast order. In this figure, we can see how the S phase begins, and this is with the creation of the pre-replicative complex, or pre-RC. Now, the pre-RC is composed of a few components. The first one is called the origin recognition complex, or the ORC, and we can find this at specific DNA replication origins. In the DNA, there are actually multiple or many DNA replication origins, and this allows the cell to efficiently replicate our DNA. The ORC recruits another protein called CDC6, and this helps load the DNA helicase onto the ORC, and together those two form the pre-RC. Now, the pre-RC has to be activated, and the one that does this is the SCDK complex. When the SCDK complex is active, it then in turn activates the DNA helicase, and the DNA helicase then unzips the DNA and prepares it for replication. At the same time, this complex also promotes the assembly of replication fork proteins like your DNA polymerase. Meanwhile, it also prevents re-replication by inactivating CDC6 and ORC by phosphorylation. And by doing this, it ensures that the DNA is only copied once throughout the whole S phase. Next, let's talk about a way that we can pause the S phase. And here, this is dependent on your mCDK. You see, in order for the S phase to go into the M phase, the mCDK must first be activated by another protein called CDC25, which is an activating phosphatase. However, when DNA damage is detected in the cell, this prevents or inhibits the production of CDC25. So unless the cell is able to repair that damage, the cell cannot move into the M phase. Proceeding to M phase, again, this is the most exciting part of our cell cycle. And there are two events that happen in this cycle. First, you have mitosis, which is the accurate segregation of chromosomes. We will not be discussing the different steps of mitosis in this video, but please look forward to a future video on that. We also have cytokinesis, in which one cell divides into two daughter cells. Here we can see an overview of the different processes that occur in the M phase. You have mitosis, which is composed of the prophase, prometaphase, metaphase, anaphase, and telophase, and it's followed by your cytokinesis. In this figure, we can see some cytoskeletal mediators of the M phase. These cytoskeletal mediators were discussed in a previous lecture. These include the microtubules that form the mitotic spindle. These structures pull apart our chromosome and segregate them equally among the two future daughter cells. Now, when those two cells are ready to divide, then another cytoskeletal element forms, which is called the contractile ring. This ring keeps on getting tighter and tighter until it divides these two cells. Finally, let's talk about how the cell can start and end the M phase. This is actually very straightforward because it only depends on one cyclin-cdk complex called mCDK. From the previous slides, we learned that in order for mCDK to work, it first has to be activated by the protein CDC25. Once it is active, it creates a positive feedback loop, and this creates even more CDC25. And this keeps going on and on until it drives the cell abruptly and irreversibly into the M phase. At the same time, the active mCDK also shuts down Wee1, which is an example of an inhibitory kinase. After a delay though, or after the M phase has completed, it is this same complex that activates the APCC, and the APCC tags our M cyclins for destruction, and in turn, this ends the M phase. Alright, so that is our lecture for today. If you would like to learn more, please check out these two references. Thank you very much, and we hope to see you in the next video.